Welcome. Thank you so much for coming and spending your
nice.
evening with
Yeah.
us. I know so many of you personally, friends of the company, friends of Craig's. And again, welcome. Many of you are our long-term clients, and we're so grateful. I have the privilege of introducing my good friend and colleague, Dr. Craig Venter. But before, I've got a few intro slides, and then I'm going to pass the baton to Craig. But I did want to just say some remarks about kind of the longevity field and precision medicine in general. And some of this many of you are evangelists in our space, but it's worth noting this slide. And we often introduce our company with this slide. And it's an unfortunate statistic because many of you know that in the US, we invest 20% of our GDP into healthcare. So that's 20 cents on every dollar. And you would think for that massive investment, by the way, that's much more than any other country on the planet, European, Asian countries. We invest far more in our healthcare system than anywhere else on the planet. You would think for that massive investment, we'd have this awesome return. And in fact, the opposite is true. We actually have neither. So we've got this massive investment, and over the last 3 or 4 years, our life expectancy has been declining. Not staying the same, certainly not going up, but actually declining. Used to be about 79, and now it's 77 and going down, not up. And that actually preceded COVID, so we can't blame COVID. And you have to ask yourself, why, despite that massive investment, is our life expectancy declining? And it's because we're investing in the wrong place. We're investing in end-stage disease, sick care. And I often say, look, if you've got end-stage disease, the United States is the best place on the planet to go because we're great at stage 3, stage 4 cancer, end-stage cardiovascular disease, and so on. But we're really poor at precision prevention. In other words, understanding what your risks are pre-symptomatically for disease, getting ahead of those risks, mitigating those risks, so you never get the disease in the first place. That's what we're really poor at. And by the way, you don't have to trust me or believe me. You all know these folks, and every day, every week, there's someone in the press, the British Royals most prominently recently. But folks like Bernard Tyson, who was the CEO of Kaiser, didn't even make it to age 62, died of a preventable massive heart attack. Steve Jobs, we all know Steve Jobs, CEO of Apple, and he didn't make it very long either, died of preventable pancreatic neuroendocrine tumor. And in our clinic here in San Diego, we found 2 pancreatic neuroendocrine tumors at stage 1. Chad Boseman, Black Panther actor, didn't make it to his late 40s because he had preventable colon cancer, almost certainly has some kind of genetic predisposition to colon cancer. So these are all just personal, public examples of the failure of our healthcare system. But there is a solution, and we've been around for 10 years, and Craig's going to speak to that. I think where we're really different is if you have to kind of give an elevator pitch for what we do, it's how do we use data to give you another 10 years of longevity, but not just longevity and health span both. How do we use that same data set, imaging data, genomic data, wearable data, blood data, to not only give you 10 years of health span, but also to give you 10 years of vitality and performance. And so we can use that data to understand your risk pre-symptomatically for common chronic age-related disease so you can get ahead of it and mitigate that risk. That's really what we're all about. And you all know there's a lot of noise in the longevity space. There's a lot of copycats out there. But we're the original company in this space, been around for 10 years. And we've published our data. So we are evidence-based, and many of the other companies in this space cannot say that. This was a recent publication of ours. And we looked at the first 1,200 clients that came through our assessment. Some of you in the room actually may be a part of that. And much like yourselves, these were all folks who came in presumed healthy, maybe somewhat arrogantly thought they were healthy. And in fact, 1 in 50 had early stage high-grade tumors they didn't know about. 1 in 50 had treatable, actionable brain and aortic aneurysms they didn't know about. And the list goes on and on. 1 in 6 with rare genetic mutations like BRCA mutations. And if you know about these findings well in advance before presentation, it turns out there's a lot you can do about it. So the key to all this is we're making these findings sort of early when it's actionable. And the overall takeaway from the paper is basically that of these presumed healthy individuals, 40% had clinically significant disease. And get this, 14% had life-altering findings that were early and actionable, like early-stage high-grade tumors, actionable and treatable aneurysms, and so forth. So I wanted to just end with a couple slides. Many of you, of course, know our San Diego clinic, but you might not know that we have a sister clinic in San Francisco. It's actually in South San Francisco. It's really about the same configuration, 12,000 square feet with MRI scanners, all of the diagnostics, beautiful client rooms. So if you're in San Francisco, please check us out. And of course, it's our goal and mission to expand our footprint, probably Los Angeles next, here on the West Coast, and over the East Coast after that. But before I introduce Craig, I'm just going to end with this slide, which is really a walk of our 10-year journey. And again, there's been a lot of folks in the longevity space, especially recently, and none of them have been around for 10 years. You know, you had Airval, which unfortunately went bankrupt in 2015. You had Google Health, which despite billions of dollars of investment, closed its doors. IBM Watson Health was spun off. And so a lot of companies really trying to reduce genomics-based medicine, AI-enabled medicine into clinical practice. Not only have we done that, but we've been around for 10 years. And Craig's going to speak to a little bit of this journey, but I think some of the important facts in our journey were that started in 2015 here in San Diego. We've got a sister clinic up in San Francisco. We've invested very heavily in our bioinformatics platform. We like to say that longevity care, which is what we practice at scale, is really value-based care. What is value-based care? It's improving outcomes while controlling costs. You can't do that without a platform. You can't do that without AI. And so all of that is part of the platform that we've invested very heavily in. We also have a very robust partnership with Mass General Hospital. And we're in fact working on a Middle East partnership with Mass General as well. So with that, I'll turn it over to my great friend, Craig, who's going to talk a little bit about our 10-year journey, but I think is also going to say a few words about his amazing book.
Well, thank you, David, and congratulations on 10 years. You've been there essentially the whole time and made it happen. So, because I was spending so much money on research, they suggested I should go back to my research institute. So,
well, thank God you did.
Yeah, but we spent in the first 5 years we spent about $400 million on research. So it's sort of the basis of a lot of things that went forward. But I thought I would just give people a brief history of how all this came to be. You saw the slide about the human genome. It's hard to believe that was 26 years ago now. My goal was to find ways to better interpret the human genome. The biggest surprise when we sequenced it, that there were only 20,000 genes. I've often argued that what are considered some of the biggest advances in science are just proving that the previous ideas were bullshit. People were arguing there had to be 300,000, even more human genes, so there'd be one gene for every human trait. It's amazing how these ideas permeate science, but so the world was absolutely shocked when we showed there was only 20,000, roughly the same as a fruit fly, and certainly the same number as your dog, your cat, your pet pig, everything else around. So it's the uniqueness of the combinations in the combinatorial level that's important. And that's hard even for human geneticists to grasp. So most of them are still in the linear world. Studies that are done trying to associate diseases with the genome are are done by studying groups of patients with the disease. So trying to interpret the human genome is very difficult. Geneticists look at populations with diseases like Alzheimer's disease, and then they look for variations in the genome that correlate with that. So there was a gene called ApoE, maybe it's your computer, that did seem to correlate well with Alzheimer's disease. Primarily with people that had 2 copies of the variant. Because my genome was the first one sequenced on the planet, lots of people studied it, and multiple labs told me that I had a variant in the ApoE gene, indicating that I had an increased risk for Alzheimer's disease. So, wanting to do correlations, I asked my friend who was head of neurology, and the other friend who was head of radiology, if I could get a brain MRI and a brain amyloid PET scan. And they said, no, it would be unethical for us to do that because you don't have any symptoms. And they're also worried that because of my genetic code and the reports that were out there, that in fact they did these tests and found that I had early onset Alzheimer's, it would make it very difficult for me to raise money or recruit people to my institute and things. Finally, I convinced them to do it. They said they would do it under a pseudonym. So I said, how about Charles Darwin? And I said, no, that's too obvious. They made up some Mormon name like Joseph Smith or something. I can't remember what it was. And, but they were very surprised. They called me a couple of weeks later to tell me that I had a brain of a 40-year-old and the amyloid PET was totally 0. My wife said there must be some mistake. They said he has a brain of an 18-year-old. But that was the first clear-cut realization of the genome as described not being as predictive as geneticists were claiming it to be. Since then, we've had a lot of heterozygotes go through HLI, and essentially they've been 100% negative. And it's only the people that have 2 copies of the ApoE gene, it's about an 80% correlation with Alzheimer's. It's not 100%. BRCA1 and BRCA2 are the same. Physicians totally misled their patients by telling them that they had a high 90% chance of breast or ovarian cancer if they had mutations in BRCA1 or BRCA2. The real data is you have a 50% lifetime risk for cancer, not high 90%, unless you have a very intense family history where every woman in your family has breast or ovarian cancer, aunts, grandmothers, which means BRCA1 and BRCA2 are not the cause of breast and ovarian cancer. They're linked to it in some way, and it means we still don't know the genes that actually cause breast and ovarian cancer. But Angelina Jolie was a perfect example of somebody with an incredible family history and opted to get a double mastectomy as prophylactic treatment. So I decided that we needed to do intense phenotypic screening to relate back to start to understand the human genome. And the direction NIH has gone in for the last 25 or 30 years has taken us nowhere, other than the good news is it's gotten orders of magnitude cheaper to sequence a genome. The first genome I did that resulted in that picture at the White House cost $100 million. Now, some of you could probably afford that, but probably not too many. And you'd probably just buy another yacht if you could. So Now it's down to one company is doing it for $200, and others are in, it's clearly less than 1,000. But the definition of what a genome is also changing now. So when we sequenced my genome, because there was no reference, we had to assemble it de novo. And that was the first time it was done in history, and almost the only time it was done in history, because Illumina developed this sequencing technology that was fast and cheap, but they could only read little pieces of the genetic code. And so the way people work out what your genetic variants are, they line these little pieces up next to my genome or one or two other reference genomes. So you get to find out how different you are from me, but it doesn't tell you how unique you are. So the technology has changed enough. We're going back to the original of being able to do complete diploid genomes, not just haploid genomes. So we will have the chromosomes that you inherited from your mother and your father. We found only 25% of all human variation is in what people have been measuring with the single nucleotide polymorphisms. So the increased complexity is going to help us answer a whole lot more questions. And but we just started HLI to do phenotype screening to understand the human genome on apparently healthy subjects. And the paradigm changed almost instantaneously. Our first client through was actually a physician, and we found he had a major tumor under his breastbone that he had no idea that he had. From there on, just every day we made new major discoveries in healthy people while just trying to collect phenotype data to understand the genome. And that is how the entire paradigm developed that you've all have experienced or hopefully going to experience. And David was giving the overall statistics. They're more important. The average age here is, I would say, above 50.
Thank you.
For people over 50, 5% of you will have a major tumor that you don't know about. And you mentioned the aneurysms. 1% of the population has brain aneurysms that they don't know about until they have a massive bleed and die. It's treatable in 15 minutes as an outpatient, but the population is skewed to those under 60. So it's genetic selection in action. People with brain aneurysms don't make it past 60, they die from their aneurysms. So that's why we see it much more in younger populations. So the paradigm changed to pre-symptomatic testing. But the medical community is almost completely opposed to this. And it's a very interesting history. It started in the 1970s when CT scanning was developed, and storefront clinics opened around the country offering whole body CT scanning. But CT is just X-ray. And X-ray, unlike the technology that we have here, cannot distinguish between a cyst and a tumor. And so when all these primarily cysts were discovered, they had to be generally biopsied. There's always additional care. The insurance companies balked at it, and they started a term, they're called incidentalomas. Medical students today are taught that it's essentially unethical to do screening tests on asymptomatic people. We're showing, in fact, it's unethical not to do screening tests on asymptomatic people. We all try to do something to control our health. But I argue you can eat 12 pounds of kale a day, exercise, but you don't know if you have a tumor that's been growing for 3 years, because until it gets big enough or metastasized, you don't have any symptoms that you have that tumor. So David finds them at stage 0 and stage 1, or sometimes a little bit later stage. I was testing the new MRI after being told, after multiple biopsies and MRI at UCSD, that I did not have prostate cancer. So I got out of the MRI, and David had the job of telling his boss that he had 2 high-grade prostate tumors that just were starting to grow down the vas deferens. They were re-biopsied and shown to be very aggressive tumors. So I had surgery right away, came back to work a few days later. But we cultured my tumors, and they grew 10 times faster in the presence of testosterone, which I was put on clinically because I had low testosterone levels. And it turns out, those who know me, and certainly if you knew me when I was 18, would say I had way too much testosterone. My girlfriend's father definitely concluded that, and threatened to shoot me. But we can tell from the genome, looking at the testosterone receptor, how much of the receptor you're going to express. So some people, it's a triplet repeat. Some people with multiple copies of that repeat are sterile. They can have all the testosterone in the world, they'll still be sterile. I have the least number of repeats known so far to science. So I massively overexpress the testosterone receptor. So my body adapted well. It only takes very tiny amounts of testosterone to get maximum activation. So I was bald at 21, always had an aggressive personality. But endocrinologists looking at that bit of data said, oh, you have subclinical levels, we have to boost them. And that was like pouring gasoline on probably some latent tumors that I had. It was just happenstance that I was doing that. Had I not done that, I would have died sometime in the last 6 years. And that's part of the longevity equation. Not doing that, now I have 10 to 30 or more years to be productive with. And we have literally now hundreds and hundreds of stories like that. So the problem is HLI works well primarily for Northern European ancestry people that have wallets thick enough to afford to come here. That's a pretty small part of the population. So at the Venter Institute, which is a 501c3 organization that all this started out of, we're taking a parallel approach. We're proposing to Arbor H to start a 100,000 person clinical trial of the exact paradigm that you can get here with 5 mobile 3T MRI clinics and cardiac clinics for populations that, for a variety of reasons, couldn't possibly go through HLI. And we're doing it as a clinical trial to test pre-symptomatic testing, the paradigm that HLI originated, to get it so it's back part of the standard of medicine. My calculation is it would save the US government $2 trillion a year if this approach was broadly used. But health insurance companies and the entire economics of healthcare is based on people getting paid for investigating symptoms. So it's not a matter of just changing people's minds, showing a new paradigm. To save that $2 trillion a year, you have to turn the whole system on its head. And the only way to do that is with data. So that's what we're doing. We have another proposal to do this for indigenous populations that have about 10 to 20 years shorter lifespan than the rest of the country. So if any of you are interested in seeing broader applications of this for the population, meet Jill Mullen. She's head of our group at the Venture Institute that takes donations and makes sure people get their tax deduction for them. So if you want to help this paradigm expand, that we think will only help HLI if it becomes the standard of healthcare everywhere. So that's all I was going to say, other than it's amazing what this has turned into in 10 years. Close to 8,000 people have gone through, some of that's counting multiple visits. We don't have an exact count on the number of lives saved or the number of expanded productive years of lifetime, but it has to be in decades. And it's something I'm incredibly proud of and proud to be associated with, and congratulate David and Wei Wu and the team here for keeping it going.
Thank you.
So now I'm going to tell you a related but different approach. It's using genome sequencing to characterize the environment. And I've always been a sailor. And after I finished sequencing the human genome, I had nothing to do for 6 months, and I had to kind of retool my brain. So I sailed down and lived in the Caribbean for 6 months, and just sailed around and really enjoyed myself. But while I was there, I started 3 new institutions, so I wasn't just goofing on. But when I was there, I read a study saying how little diversity there was in the oceans. And this just didn't make any sense to me at all. Bermuda, we were talking earlier, some of you have sailed to Bermuda or been there in other ways. It's part of the Sargasso Sea, based on the seagrass there, but it's considered by science to be an ocean desert. It's thought that there's so little life other than in the Gulf Stream. It just did not make any sense to me at all. So I proposed a simple experiment and proposed it to the Department of Energy to fund. And every reviewer turned it down with extreme urgency. And only the director of biology at DOE, Ari Petrinos, believed along with me that computing and biology were the future of science. And so we just tried taking a barrel of seawater, filtering out all the microorganisms, and then sequenced them just like your genome was sequenced. And the algorithms that we developed early on ended up being very powerful. And that's one of the things I learned at the earliest part after we sequenced the very first genome in 1995. That each species and each one of you has a unique mathematical solution for your genetic code. That's why we can independently assemble each person's genome. And so I thought we could take very complex mixtures of DNA, like just chopping up everybody in the room and isolating the DNA and sequencing it, we'd be able to reconstruct each one of you. And basically that's what we did in the ocean. Organisms were numbered one at a time as they were discovered in the Sargasso Sea. They were up to SAR 11, Sargasso Sea 11. So it was the 11th microbe. You have to understand, before I brought in genome sequencing, the only tools that microbiologists have had for the last several hundred years was getting things to grow in culture and looking at them under a microscope. So we didn't want to look at the organisms, we didn't want to culture them, we identified them just by sequencing their genetic code. And just from the one barrel of seawater, we discovered thousands of new species. We stopped sequencing after 1.5 million new genes were discovered. And it's a paper that sort of blew everybody's mind in the field and changed all the thinking overnight. That's what every scientist hopes to have, is a one paper that changes how everybody thinks. But again, it's just because they were stupid how they were thinking. It's not because I was so effing brilliant. But it just shows you a simple idea where you can go out and characterize the environment and find out how little we know. So this expedition, we followed the route of the Challenger, which was in the 1870s, was the first actually sailing expedition. Just lost some lights, but that's okay. And again, it was based on a faulty scientific notion. Scientists at the time were sure there couldn't be any life below 1,800 feet in the ocean. So the Challenger, and it was when first transatlantic cables were being laid, we wanted to understand what the sea bottom looked like. The Challenger stopped every 200 miles, sent a dredge to the bottom, pulled it up, and every place they did that, they discovered new life, new organisms. They discovered the Mariana Trench, the deepest parts of the ocean. They found life everywhere and sort of blew that previous theory apart. But they could only base things on their visual acuity, so they could see macroorganisms. And that's the same thing with Darwin. Some of the water we've been in, it's crystal clear. You can see for hundreds of feet. You can't imagine that there's anything in it. But every milliliter of seawater around the world has 1 million bacteria and 10 million viruses. Doesn't matter how crystal clear it is. Think about the next time you're in the ocean, you swallow a mouthful of seawater, how much unique biota you just consumed. The fortunate thing is very few of these are human pathogens. And so we set out, like the Challenger did, to stop every 200 miles, filter everything from a barrel of seawater, ship it back to the lab, and have it sequenced. The amazing thing was every 200 miles, 80% of all the sequences were totally unique. So one possible thing was the ocean was a giant homogeneous soup, which is what most people assume. In fact, it's millions or tens of millions of microenvironments. You can have wind blowing, more oxygen, no sunlight, so many different things affecting each environment. But it shows, we calculated the diversity of microbial life, and it helped us recruit astronomers and physicists, because we have about over 10,000 times more microbes on this planet than there are stars and planets in the universe. So they decided it was far more interesting to count microbes than to count stars. It just shows how much diversity there is. And this is a microbial planet. We sequenced the very first microbiome. You all have about 3 or 4 pounds of microbes growing in and on you. They're part of having healthy skin. You have a different set of colony, different microbes around each tooth in your mouth. And the people that study these are arguing dentists that, who try to kill all the bacteria in your mouth is being the totally wrong approach. There's some microbes you do want to get rid of. Most of them are protecting you. So you're a microbial host to about 100 million bacteria and 10 times that in terms of viruses. You can't find an environment without microbial life in it. Even at the extremes, So the highest temperatures are about 140 degrees centigrade. That's boiling water is 100 degrees centigrade. We find organisms that live in very high radiation, pH that's so low or so high, if you put your finger in it, it would dissolve. So the diversity here is amazing, but the key thing, the microbes in the ocean, in the top layers, produce about 50% of the oxygen we breathe. And we're very essential for terraforming planet Earth. Some of those microbes got incorporated into larger cells that became plants, but it's the same basic mechanisms. So all of our oxygen, what created the oxygen atmosphere on Earth, came from these microbes and subsequently plants. So we don't want to destroy these microbes in the ocean, but we seem to be working very hard to do that. There's now anoxic zones, no oxygen in these zones. The largest patch is the size of the continental US. There's been a huge one off of Oregon. Fish can't live in there. If they go in there, they die because there's no oxygen. This is happening to rivers and streams in England. And what they do, they actually pump oxygen into these streams to keep fish alive, because fishing is an important part of their tourist industry. We can't do that with the ocean. If we screw this up, we're gone. The microbes will still be here, various types. They adapt well. And we're sailing in different places, sailing off of Costa Rica. There's multiple currents that converge. We sailed into a 5-mile wide patch of plastic and trash. There's actually a refrigerator floating there that the crew members wanted to open and see if there was any beer in it. But, these have now grown quite substantially, and they get broken down into micro pieces of plastic that basically everyone on this planet now has these microplastic particles circulating in your bloodstream, and nobody knows what the impact of these is going to be. So it's not just CO2. Changing the ocean temperatures, even 1 degree. I'm sure many of you have been to the Seychelles and you've seen the beautiful white sandy beaches there. Those are a result of a 1 degree centigrade increase in temperature in the Indian Ocean, killing off a microbe that was a key symbiont for keeping the coral alive. The coral died, bleached out, degraded, and became those white sandy beaches. So one of the largest living organisms on our planet was killed by 1 degree change. This is a problem now on the East Coast. I spent summers in Maine. The temperature is slowly rising, making it so that the lobsters and other key species that are worth billions to the economy are migrating north to colder water. So the Canadians like it, but it's not helping the rest of the world, and it's just a matter of time till we lose some of these species altogether. So it was a voyage of discovery. We sailed 65,000 miles, but it also had its entertaining moments. I did an interview with a reporter from Nature magazine who read the book, and she said, it seems like you had a very large number of near-death experiences. And I said, yes, but they weren't planned to be that. So we were arrested twice, first by the French government and then by the British government. When we sailed into the Marquesas, the French arrested us, threatened to sink our boat if we left harbor. which we took seriously because they sunk the Rainbow Warrior, the Greenpeace boat in New Zealand harbor for protesting their nuclear testing. We had to get permits through the State Department for every country's waters that we tested and sampled in, just taking a barrel of seawater. And we got caught up in local politics. So French Polynesia government was claiming they were in charge of their waters, and Paris was claiming France was in charge. So we got caught up in their internal battle. We had to involve the State Department, and the US Navy intervened, telling the French Navy that the Sorcerer II was a US research vessel and under US Navy protection, and to back off. Partly what the French were worried about is that I was going to sample near the atolls where they did all the nuclear testing, which would show how much environmental damage they did. But it's far too radioactive. I wouldn't have gone there even if they said okay. When we spent 6 months in Brisbane and around Australia waiting out hurricane season, and I flew back and forth to the Institute to keep it running. I flew out for all the ocean passages, and I just flown into Australia, gotten on board the boat in a marina in Brisbane, took my shoes off, was having a beer, and I saw this SWAT team boat coming up the river. I thought, gee, that's interesting. I wonder where they're going. And then they pulled in to the marina. I thought, oh, that's even weirder. And then they pulled into the slip next to Sorcerer, and I thought, this is something is going on. And they came out with their weapons drawn and said they wanted to board Sorcerer because they've been told that we had a meth lab on board. I said, well, we got to finance the voyage somehow, I said, well, we're a U.S. research vessel and you don't have a warrant, and I do not have to let you in without the State Department being involved. But I knew that would just be trouble, and when the Coast Guard and other parties board boats looking for drugs, they basically tear the boats apart looking for them. So it was a woman, a captain, head of the SWAT team, and I said, I'll make a deal with you. If you all take off your combat boots, holster your weapons, and if you want to see in any compartment, you ask a crew member to open it, I'll let you on board to check. Meth labs apparently have a very strong odor. She found out very quickly it was a luxury 100-foot yacht and not a meth lab, and they were kind of embarrassed. And then we learned the true story behind it of apparently the person that called was the former fiance of one of our crew members, Jeff Hoffman's current girlfriend in Brisbane. And this guy was so upset that he got dumped by this girl for a Sorcerer crew member that he called the police and said that we had a meth lab on it. So, it turns out the SWAT team actually posed with us for photos, and I asked them if they would arrest Jeff and take him away, but they said they wouldn't. So that one turned out a little bit simpler. When we left Australia and were sailing across the Indian Ocean, there was this beautiful set of islands about halfway across called the Chagos Atolls. That's where Diego Garcia is, our B-52 air base. And there's this small island called Chagos that supposedly has the best inner reef and life of any place in the world, because the British removed all the natives and sent them to Mauritius because of using them for military bases. So they let sailors stop there on the way across the Indian Ocean. I notified the State Department we were going to stop there and we weren't going to sample. And we weren't there very long until this another rib approached us. There were no insignias, no flags, just 12 heavily armed people came up and said they were boarding us. I thought about resisting them, but we had a 12 gauge shotgun, an M14, and a 22 mag pistol. So I don't think we would have lasted long. So they boarded us, took our passports, put us under arrest, their concern was because of my notoriety and the expedition's notoriety, we were going to bring attention back to the Chagos Islands and what the British had done to all the natives. So somehow they thought it'd be a really good idea to arrest me. It just so happens from all my interactions with President Clinton and his team, there's a thing called Renaissance Weekend. We used to spend every New Year's with him for several years, and the person who runs that is Phil Later, who the president appointed to be the ambassador to the Court of St. James, to the UK. I happen to have his personal cell phone number for some reason on me. And I had a satellite phone in my cabin, and I called them and explained what happened. That people with no insignia, no identification, but claiming they're British military, took our passports and are holding us at gunpoint under arrest. And he said, I'll get right back to you. And he immediately went to the top of the British government, and he said that they're going to come back again with a different approach. They came back in full battle dress with a full, battle British flag and dress uniforms, but still had their weapons drawn. And they had a proposal for me. They said if I signed this statement that I had broken a law, they would return our passports and let us go. And the law that I supposedly broke was having, it's an 18th century law that Britain had to give permits to sailing vessels that had dive equipment on board. So this is all they could come up with, some law from the 18th century that we didn't have a permit from the British government for having dive equipment on board. The captain was standing in my cabin with another person with weapons drawn. I had the ambassador on the satellite phone, and they presented this statement for me to sign and said if I wasn't willing to sign it, they would leave it with me for a week and come back and see if I was willing to sign it, and they would keep doing that until I was willing to sign it. So I said, Mr. Ambassador, I'm going to sign this agreement, but I'm signing it under duress. The military people had no idea what that meant. So I signed it, they took it and left. They refused to give me a copy, even though I said I couldn't talk to the press or disclose it. And gave us our passports. We pulled up anchor. We just got about a mile outside the atoll, and the satellite phone rang, and it was the US ambassador, and he said, stop or they'll sink you. I always take things like that somewhat seriously. So we stopped immediately, and I said, what possibly is wrong? He said, the British government won't accept an agreement signed under duress. So I want you to think very carefully before you answer this question. Were you under duress when you signed that agreement? And the only thought going through my head was, I'm really fucking under duress now. But I thought for a full moment, and sometimes I can be pragmatic. This surprises people. And I said, Mr. Ambassador, I was not under duress when I signed it. He said, that's what I was hoping you would say. You're now free to go. That was the end of it, except so there's a British version of the book that just came out, and this incident is described in detail in it. The British editor said she read the book and really enjoyed it. And I said, you didn't have any trouble with me describing the British government arresting me and doing all this. She goes, I must have skimmed over that part. So I thought they were going to remove it in legal review. The only thing they cared about in the legal review was the Brisbane incident, that we changed the name of Jeff's girlfriend so she couldn't be identified. So they were more focused on that. So the British government arresting us falsely under gunpoint didn't matter. So you sail around the world, you're going to have adventures. They're not always what you expect them to be, but we discovered more species in those 65,000 miles than the rest of the history of science combined. And any child can take a cup of water to a river, a stream, the ocean, get things sequenced in it, make brand new discoveries that exceed what most people have done in the past century. One of the questions that I always get with this, you keep showing, what we don't know. What else don't we know? And I said, there's no way to answer that question other than people doing what I do, go out and ask simple questions about the world and try and get answers for them. So thank you for your time and attention. I'm happy to take your questions, and then we have some books for you. Yes, sir.
The Greenland ice borings go back 10,000 years, and they talk about the temperature of the Earth during that period of time. It was much higher than 10,000 years ago. It comes down, goes up and down, and we've just changed direction to go up again. So it just seems to be something that's so much bigger than all this stuff that we talk about with global warming. I just would like to know what your comment is on that.
So I don't know if people could hear you. Talks about this sort of the natural cycle that happens over millennium with temperatures on the Earth. But when you look at history, there's a totally different range of life. Originally no life when the Earth was extremely warm. Then the photosynthetic organisms developed and we got an oxygen atmosphere. Life evolved to get mammals. The environment is constantly changing. And when the environment changes, it also changes life on the planet, whether it's from an asteroid strike that got rid of all the dinosaurs or other things that have been fairly drastic changes. So we're dealing with a situation that was stable for our condition before we sort of overpopulated and overpolluted the planet. Everything's been based on natural cycles, not human behavior and human chemicals and conditions. So we're in a totally new world. And just because there's been temperature fluctuations in the past, it didn't matter when there wasn't mammalian life. Maybe we lost some microbes, maybe we grew some new ones. So we're in a unique position now with 8 billion people that look more like you than dinosaurs, for the most part. And learning the consequences of individual action is a tough thing for everybody. Lots of people change their motor oil. This is probably not a group that does it themselves, but I did as a kid, and you never thought anything about just dumping it in the ground or dumping it in the water. You think your one action has very little impact on anything. You know, now we're faced with all of humanity in Tijuana putting essentially all the raw sewage and flushing out the Tijuana River that's now polluting all the way up to the La Jolla beaches. And causing human disease and causing problems along with the anoxic zones. You know, we are changing our environment faster than any natural cycle. Other questions? Yes.
First of all, that was one of the most brilliant answers I've ever heard about the whole picture. Thank you.
Thank you.
All the species that you found, more than in the universe, right? One of the things you've done is sequence them. And I understand that the difference between them are a lot larger than the difference between the normal sequence of all of us together. Can you talk to that in terms of the significance of it and what that can ultimately help us discover?
Yeah, so one of the things, for example, all these microorganisms create much more complex chemistry than our best chemists can produce. Microorganisms are sort of in biological warfare with each other. That's where a lot of antibiotics came from. They create an antibiotic that kills off, somebody they're in competition with. So the wide range of new biosynthetic pathways, these billions of organisms, if those are harnessed, it will create totally new chemical libraries for pharmaceutics, for industry, for building, the kind of things people are looking for of organisms that will create all the building materials on Mars instead of trying to, send billions of tons of building materials up there, getting microbes to grow it and reproduce it. So the potential there is enormous, and that's what the whole field of synthetic biology and the synthetic cell we made is all about. And it's an area that China is outspending the US 10 to 11 to 1 right now. Last year they put $11 billion into this space versus the US's $1 billion. And there's a lot of concern at the top levels of government of somehow trying to block that and increase the competition. So these microbes could be our future in more ways than one. Yes, sir.
Did you do any exploration in either the Arctic or Southern Ocean?
So we didn't sail there, but yes, the Venture Institute we have an expedition teams that go down almost every year, sometimes twice a year. And one of the most interesting things is we sampled from a frozen lake that had been sealed off, and we found about 10 times higher density of life in the frozen lake than in the open ocean. So people expected they thought it was going to be a sterile environment. It was just the opposite, that 10 times the organisms that the ocean had. So, and Antarctica is, it's interesting, same with the permafrost, all the things that are frozen from past history. So we can tell what the past climates were. We can even come up with ancient microbes. Part of the worry is people that died from the plague, people that died from smallpox and other diseases are buried in the permafrost. And as that starts to thaw, there's concern about resurgence of these diseases. So, right in the back.
With regard to the
Really shout so I can hear you.
I got you. With regard to the anoxic zones, how deep down into the ocean do they go, and what sorts of microbes did you find if you sampled the anoxic zones in the ocean?
So I I'm not sure how deep they go, but you don't have to go that deep to get out of having any oxygen anyway, and not no sunlight. So the world, our planet is full of anaerobes. We've sampled in mile deep lines and the Colorado coal bed methane mines and things, and we found tremendous diversity. But microbes that we found a mile deep in Colorado, close to 100 degrees centigrade, no oxygen. They live off of sulfur and other metabolites. The closest relative we found was from a volcano in Italy. So some people think there's more life subsurface in the earth than there is on the surface or even in the oceans. And as I said, we found tremendous diversity, but it's a different kind of diversity than we have in the oceans. The biggest drivers of evolution are sunlight and oxygen. Both are very strong mutagens. So these organisms that are photosynthetic in the top 30 to 40 feet of the ocean are exposed to UV light. They turn that into energy and oxygen, and also produce a lot of oxygen. So that's why there's such constant evolution and such tremendous diversity. And there's neither of those in the deep earth. So it's totally oxygen-free zones, but we're in a microbial world. Some of these organisms, like the radiation-resistant ones, can have been detected on the surface of the space station. An experiment, Japanese did a coating. They lasted over a decade, and they were still happy there in space. I got a former head of NASA upset with me when at a press conference. I said the outside of the space station is covered in feces, which he didn't deny. He just didn't like me saying it. So what they did early on in the space station, they just pumped all the human waste outside the space station. And a lot of those just settled on the surface of the space station. Now they package things into stainless steel containers and launch them back into the atmosphere. So I tell people to be very careful about wishing upon shooting stars. I might choose the wrong one. We got time for a couple more questions.
Gentleman to the left here.
Oh, yes.
Thank you, first of all, for the presentation, and I love the exploration in health. My dad died of dementia and Parkinson's 2 years ago.
I'm sorry.
So he was 79. He lived a pretty good life, but it's put me on a trajectory to find answers and create longevity for myself and those that I love. David mentioned earlier about the fact that millions of dollars, billions of dollars are being spent in reactive medicine and not so much proactive prevention. And I think that's true. And I wanted to get your opinion on how is it that what does the future look like to using the Earth's resources to create prevention, to create a healthier body for ourselves? How do you feel about holistic medicine, and can we use the Earth's natural resources to create a better life for ourselves? And where is that going? What does it look like 20 years, 50 years from now? And sorry for the long question.
So,
But
well, it was 32 questions. But, well, the best thing when there's a family history of any disease, but Alzheimer's is a particularly interesting one that, having early measurement of the brain MRI here creates a baseline that measurements can be used where you become your own control. And so you can see if you're actually changing versus your data compared to the average of everybody else that's been doing it. That's far more important because the ranges are so high. Also, genetics will tell you whether you're at increased risk. If you're homozygous for ApoE4, you're at a much higher risk than the average population. There are new markers showing up for Parkinson's, but I think MRI is probably the best detection still for that. I mean, there's new things coming up every day. One of the most interesting things that came up in science 2 days ago is that Viagra seems to help slow the onset of dementia. So,
Heard it here first.
there could be 1,000 reasons why that's so. But there is a thing called vascular dementia. That's why it's so important to have your blood pressure under control. So Viagra being a vasodilator could just be helping reduce vascular dementia. It's not clear at all. But just like the new glucagon agonists that were for diabetes ended up being phenomenal for weight loss and are now proven to be very cardioprotective. There's no idea yet what they do for the brain. It's part of what we're going to be doing with these MRIs, with the 100,000 person clinical trials, is looking at various drugs that supposedly affect longevity. But all these studies are done primarily in mice. We've shown multiple times mice are not little humans. So there's lots of research going on, but the best thing you can do is get your own baseline, get your own genetics, and don't make the mistake that Steve Jobs did.
Yeah.
He could have easily been alive today if he didn't go into holistic medicine.
Yeah.
So, if that doesn't express my opinion.
I'm going to add, Craig, just one other. We feel so strongly about this in our clinical practice here in the clinic. You know, we've learned that 40% of the risk for most chronic age-related diseases, including dementia and Parkinson's, is modifiable and in your control. In fact, the Lancet Commission had a publication in 2020 that established just that after doing an extensive review, extensive meta-analysis. 40% of the risk for dementia, vascular, Parkinson's dementia, Alzheimer's dementia, is modifiable in your control. And it drives me crazy to no end when you hear a lot of folks in the longevity space talking about magic elixirs and rapamycin and NMN. None of it's proven, and actually, frankly, a lot of these medications have side effect profiles. And what's really important is you don't even need to resort or turn to some of these so-called magic elixirs, because we know that 40% of the risk is modifiable in your control. And really, our job as a clinic and as your doctors is to help you appreciate what modifiable risk factors are most important for you. And so if there's a laundry list of 20 modifiable risk factors, which is the number one lever to pull to help you sort of mitigate that risk? And I think that's really important, because the number one modifiable risk for all chronic age-related disease is advanced age. And we know that actually the aging process is probably the biggest modifiable risk factor. And if you think about serious neurologic diseases like Alzheimer's, like Lou Gehrig's disease, like Huntington's, very few, if any, folks get those in your 30s, right? And by the way, for Huntington's disease, which is a trinucleotide repeat disorder, you're born with that risk at birth, but you don't get that disease until 30s, 40s, 50s, 60s, not in your youth. And the reason is that age is the biggest risk factor. It's mitochondrial dysfunction, it's insulin resistance, it's inflammation, and that is all modifiable in your control. So I think part of what we try and do as a clinic is use data to help you understand what the most important personalized modifiable risk factors, and there's nowhere where that's more true than for Parkinson's. So I just wanted to leave you with that thought.
And physical activity and exercise are very high on the list, particularly for longevity. A study in women, if they had the strength and the mobility to keep moving, they lived much longer than people that couldn't get out of the chair. But you know, it makes you want to do a Viagra study to see if it's just increased exercise. Yes, sir.
First and foremost, I want to thank you. I want to thank David. I want to thank Pam, Jill, and everybody else associated with the organization. Because you guys are in the trenches and on the front lines every day doing this work. Thank you for your work and your service. That's the first question. Second question to you, Craig, is that with all this deconstructing of how all this works, you open up this world to all of us. And I wonder if you're ever lying in bed late at night thinking about other worlds. You know, Neil deGrasse Tyson tells us we don't live in a universe. There are universes. And if any of your work in the next book or in your imaginings, you think about the possibility of these combination of factors operating in the universe, on other planets, in other ways.
Yeah. Well, thank you for your kind comments and being here. In my last book, I wrote about life on other planets, and we will find microbial life ubiquitously throughout the universe. Earth and Mars exchange about 100 kilograms of material a year, and that was before rockets went there dropping things and stuff. So theoretically, you can't take a shovel of dirt on this planet without having some Martian soil in it, and vice versa. So Mars had oceans. In theory, they're subsurface now. There's a very high probability of life in those oceans. If we find it, it'll be very similar to what we have on this planet. And microbial life is what will be ubiquitous. Many people have argued that the universe is not old enough to have intelligent life develop at multiple independent points from that microbial life. I'm still waiting for it to happen on Earth. So it could be another millennium or so. Yeah.
I think we've got one more, and I think then we'll move to signing.
Do you have any comments on regenerative stem cells, like mesenchymal stem cells, helping with regeneration, lowering inflammation, and producing more blood flow and just regenerating parts of the body and disease of the body to help us as we age? Do you have any comments on those stem cells?
It's an area of tremendous hype, creating hope. It's mostly anecdotal data because most people that do stem cell research do not want to do controlled clinical trials. And so stem cell companies, including one of our co-founders, they start and they go because that's why we refuse to do them here at HLI, because they would not do clinical trials. Testimonials are very powerful, but with new drugs to treat the pain of osteoarthritis, there's a 50% placebo rate. So people have to demonstrate in their clinical trials that their drug works better than a sugar pill, which works half the time for people. So some people I know have had stem cell treatment for their knees versus surgery and swear by it. And seem to have recovered and not have the pain. But, these are all one-off stories. And the only way science proceeds is by asking precise questions under controlled conditions to separate out what we want to believe from what is. It's still open about stem cells, but we have to approach them differently, than people telling good stories.
Well, thank you, everyone.